Glycogenolysis. Glycogenolysis means breakdown of glycogen, which is a store of glucose, into glucose 1-phosphate and glucose. So when does glycogenolysis take place inside our body? First question. Whenever you are starving, during that time, in order to support the energy needs of the body, the glycogen, which is stored in liver and skeletal muscles, we call them hepatocytes for liver and myocytes for muscle cells. The stored glycogen over there gets converted into glucose 1-phosphate and glucose. Hello, my name is Dr. Kamya Tikku, and today in this video, we are going to focus on a portion of glycogen metabolism, which is called glycogenolysis. Just like any other kind of metabolism, glycogen metabolism also can be divided into two parts, which is glycogen catabolism, that is glycogenolysis, and glycogen anabolism, that is glycogenesis. Glycogenesis or glycogen anabolism takes place inside our body when we are in well-fed state. That means we are having our food properly and we are able to store that carbohydrate also into the form of glycogen. Whereas on the other hand, glycogenolysis takes place when a person is starving or fasting due to any reason. Since we know that blood glucose is a source of energy for the entire human body, whenever a person fasts, to maintain normal blood glucose levels, the liver plays a central role in producing glucose via glycogenolysis. There is one more pathway which works parallel to the glycogenolysis pathway, and that is called gluconeogenesis. I have already discussed this in my previous video. You can watch the video. I'll put the link of this video in my description box. First of all, let's understand what is glycogen. Glycogen is a storage polysaccharide consisting of D-glucose residues. It is made up of alpha-1,4 linkages, that is linear chain, and alpha-1,6 linkages, that is branched arrangement of D-glucose. This highly branched structure of glycogen helps it in order to organize properly inside a smaller space. The advantages of the highly branched nature are the increased solubility and the ability to concentrate a larger molecule in a shorter space. Now, the liver breaks down glycogen to maintain adequate blood glucose levels, whereas muscles break down glycogen to maintain energy for contraction. The process of glycogen breakdown can either occur in the cytosol or in the lysosomes. When glycogen branches break down, the first enzyme which acts upon them is called glycogen phosphorylase. Glycogen phosphorylase converts glycogen branches into glucose 1-phosphate. This glucose 1-phosphate then can be converted to glucose 6-phosphate with the help of the enzyme mutase. With the help of the glycogen phosphorylase enzyme, all the alpha-1,4 linkages can be broken. But glycogen phosphorylase has limitations. It can only cleave until it is 4 units away from a branch point. Now comes the role of glycogen debranching enzyme, which will take care of the alpha-1,6 linkages. Glycogen debranching enzyme is one of the few known proteins which possess two independent catalytic activities. So the two activities are transferase activity, that is breaking down the alpha-1,6 chain and converting into alpha-1,4, and glucosidase activity, which helps in removal of the last glucose also from the alpha-1,6 branch. Now we are left with all alpha-1,4 chains of glycogen, and this can be taken care by or catalyzed by glycogen phosphorylase, and thus glucose can be recovered. Adrenal hormones such as catecholamines and glucocorticoids regulate hepatic glucogenolysis, and adenosine stimulates hepatic glycogenolysis through the secretion of corticosterone from the adrenal gland. Now what happens? The process of glycogenolysis starts in muscle due to the activity of enzyme adenylyl cyclase. Adenylyl cyclase is an enzyme which converts ATP to cyclic AMP. This cyclic AMP then binds to phosphorylase kinase and converts it to its active form, phosphorylase kinase, phosphorylated form. This activated phosphorylase kinase then converts phosphorylase B to phosphorylase A, which is the active form. And finally, it catalyzes the breakdown of glycogen to glucose 1-phosphate. During this process, glycogen synthase enzyme activity is inhibited so that glycogen formation doesn't take place. And that's all about the video. Thanks for watching.